Assalamu alaikum guys and welcome to my YouTube channel Biology Articles and MCQs. Previously we were studying about glycolysis and now we are going to start the second step of cellular respiration, that is called Krebs cycle, or it is also called citric acid cycle or TCA cycle. First of all, we will learn about that where in the cell this cycle is occurring. So the Krebs cycle is occurring in the mitochondria. But here you are seeing that there are different portions of mitochondria. First of all, there is an inner membrane and there is an outer membrane of mitochondria, and between the inner and outer membrane there is a portion that is called matrix. Matrix is actually the portion where this Krebs cycle will occur. Then how this Krebs cycle will take place and which are the products of Krebs cycle we will study next in this video. So let's start the cycle. The cycle starts from the product of the glycolysis, that was pyruvate. You know that in the glycolysis glucose was changed into pyruvate. Now this pyruvate will change into the further compound, and next the whole cycle that is called Krebs cycle will takes place and different products will form in this cycle. First of all, there is pyruvate molecule, and the pyruvate molecule will lose its carbon dioxide. Here you can see that there is one carbon and two oxygen molecule. First of all, the pyruvate will lose its carbon dioxide and will change into this compound. Here you can see that one CH3 and C double bond O is present. Next, the CoASH molecule, that is coenzyme A, along with a SH group is present here. It will enter into this group that is formed by the removal of carbon dioxide, and this CoASH will attach here in this molecule, along which the CoA will only attach its CoAS portion here, but this hydrogen will be received by NAD positive, and this NAD positive will then converted into NADH. After these all these conversion, the final product of the step 1 will produce that is called acetyl CoA. Here you can see that there is a CH3 molecule and C double bond O along with this CoAS is attaching to it. Here is the pyruvate is converted into the acetyl CoA. This acetyl CoA is now actually going to enter the Krebs cycle. First of all, we will see that there is a Krebs cycle. Here, this acetyl molecule is formed in the previous step, and now this acetyl molecule will enter in the Krebs cycle, and the first the reactant of the Krebs cycle is oxaloacetate, and it will convert into the citric acid, and then all the conversions will take place. Now now let's go. We are going towards the oxaloacetate molecule, and we will further study these steps one by one along with the structures of these molecules. In the first step, there is an oxaloacetate molecule. You can see that this oxaloacetate is carrying 4 carbons. This carbon number 1, 2, 3, and that is carbon number 4. And this is the acetyl CoA which was formed previously by the pyruvate molecule. This acetyl CoA will enter into the oxaloacetate molecule and will attach with its carbon, forming this type of molecule. Here you can see this yellow part is the acetyl CoA, and it is now attached to the oxaloacetate, converting it into the citryl CoA. Now this CoA SH will remove from the citryl CoA, and the hydrogen will also attach to with the CoASH, and this hydrogen comes from water, converting this CoASH into the CoASH molecule, and the citryl CoA will convert into the final product of step 1, that is citrate. Here you can see that there is same portion present here, and this CoA is removed and attached with the hydrogen, converting it into CoA as such, and there is C double O. This O is now entered here, converting into C double O molecule. So this is the first step. Now in the second step, there is a citrate molecule, and this citrate molecule now first of all will remove its water. The hydrogen from the carbon number 2 and OH molecule, that is hydroxyl molecule from the carbon number 3. There is removal of water, and there is now the formation of cis aconitate, and cis aconitate has the double bond between the carbon number 2 and 3 because hydrogen and hydroxyl lines are removed from it. Now again there is entrance of water molecule, and now the hydrogen of the water molecule will attach to the carbon number 3 and hydroxyl of the water molecule will attach to the carbon number 2, converting citrate into isocitrate molecule. This is only the this step is only the exchange of the carbon and hydroxyl molecules from the carbon number 2 and carbon number 3. So in the second step, citrate is converted into isocitrate. Now in the third step, there is isocitrate molecule, and first of all the conversion will take place, that there will be the removal of hydrogen molecule from here, and this hydrogen molecule will be received by NAD positive, converting it into the NADH, forming a molecule like this. Here you can see that hydrogen is removed from here, and there is the formation of a double bond in the carbon number 2. Here this is carbon number 1 and this is carbon number 2. Hydrogen is removed from carbon number 2, forming the double bond with the oxygen. One hydrogen is removed from here, and the second hydrogen is removed from here. One hydrogen will attach to the NAD positive, converting into the NADH, and the second hydrogen will exist here as hydrogen ion H positive. Now this molecule will again lose its carbon dioxide molecule, and converting it into a further product. Here you can see that there is no carbon dioxide present, and there is the formation of a double bond between the carbons of carbon number 2 and carbon number 3. Next, the hydrogen ion will remove, forming this type of molecule. Here you can see that here is same as C double bond O, and then CH2 here, and C double bond O in the carbon number 2, and there is just the difference of carbon number 3. There is CH2 is present. So this final product of this step in the conversion of the isocitrate into alpha ketoglutarate with the formation of one NADH and removal of one carbon dioxide molecule. Now the alpha ketoglutarate will again remove its carbon dioxide from the first carbon. Here is the removal of carbon dioxide molecule. But along with the removal of carbon dioxide from the carbon number 1, there will also be the addition of CoA molecule on the carbon number 1. Previously we have studied that the CoAs only will attach to the molecule and not the hydrogen ion. So this hydrogen ion will be received by NAD positive, converting it into the NADH. So the product will be the succinyl CoA. Here you can see that there is attachment of CoAs with the carbon, this carbon, and because one carbon dioxide is removed here, the product that is succinyl CoA is formed is 4 carbon compound, and the reactant was 5 carbon compound. Okay, here you can also see that there is removal of one carbon dioxide and one NADH molecule. In the next step, this succinyl CoA then add one phosphate molecule to it, to its carbon number 1. With the removal of a CoA molecule, this CoA molecule is removed, and now this phosphate molecule is entering, converting into the compound that is called succinyl phosphate. Succinyl phosphate now will lose this phosphate molecule, and this phosphate molecule will attach to the GDP, that is called as guanine diphosphate. It will convert into the GTP. GDP has two molecules of phosphate, and GTP has three molecules of phosphate. One molecule is gained from the succinyl phosphate, and the final product is here, that is called as succinate. There is no phosphate molecule in the succinate present, and the succinate is also a 4-carbon compound. Now, and in the next step, the succinate will remove its two hydrogen molecule, one from the carbon number 2 and one from the carbon number 3, and these two hydrogen molecule will be received by FAD, that is flavin adenine dinucleotide, and this FAD will convert into the FADH to converting succinate into fumarate. You can see that by the removal of two hydrogen molecules from the succinate, there is the formation of one double bond between the carbon number 2 and carbon number 3, and now this compound is called fumarate. In the next step, fumarate is now simply going to add one water molecule in it and converting it into malate. You can see that the one hydrogen of water molecule is added at the carbon number 3, and the hydroxyl group of the water molecule is added at the carbon number 2, and converting fumarate simply into malate, and this reaction is carried out by the enzyme that is called fumarase. In the next step, now this malate compound will going to again lose its two hydrogens. Here you can see that there are a loss of two hydrogen molecules, one here hydrogen molecule, and the second hydrogen molecule is this, and these two hydrogen molecule will add into the NAD positive, converting into NADH along with the formation of one hydrogen ion. Now this malate is converted into the final product of of Krebs cycle, and we also called this we have also studied this as a reactant because it is a cycle, and you know that in cycles one molecule also act as a reactant and product as well. So there is a final conversion of malate again into the oxaloacetate, and the cycle will repeat again and again. You can see that there is a removal of two hydrogen molecules, and along with the removal of two hydrogen molecules, there is a formation of one double bond here in this position. Is a formation of one double bond, and this is the final product that is called as oxaloacetate. So next we will talk about that what are the products of Krebs cycle, means how many ATPs or GTPs or NADH, FADH2 are formed all over this cycle. So let's revise this cycle once, that now we start this cycle from citrate or simply citric acid, and then end this cycle from oxaloacetate. The firstly oxaloacetate into citric acid, and again the convergence takes place along with the now and ends at the oxaloacetate again. You can see here there is formation of here one carbon dioxide. The second carbon dioxide is forming here. Means in one cycle of in one Krebs cycle there is formation of two carbon dioxide molecules. But when we studied glycolysis, we studied that there were total two pyruvates were forming. So if we consider two pyruvates undergoing Krebs cycle, there is will be the formation of double of the number of products. So the total number of carbon dioxide molecules mainly in the Krebs cycle will be 4. Here you can see that there are 6 carbon dioxide molecules written. The 2 carbon dioxide molecules were formed when the pyruvate was converting into acetyl-CoA. When the pyruvate was converted into acetyl-CoA, one carbon dioxide molecule also formed here, the second in this step and the third in this step. So when we double the molecule of carbon dioxide, there will be the total 6 carbon dioxide. Next, we will talk about the FADH molecules that are forming. Then there is simply one FADH2 molecule formed in one cycle, and the double the FADH2 molecules will be in the 2 cycles. Next is the turn of NADH. You can see, you have seen that when the acetyl-CoA was converted from the pyruvate, there was the formation of NADH also. So, first NADH here, second NADH when isocitrate is converted into alpha-ketoglutarate, third when alpha-ketoglutarate will convert it into succinyl-CoA, and the fourth one is here when in the last malate is converted into oxaloacetate. So the total 4 NADH molecule will form in one cycle, and 8 NADH molecule will form in 2 cycles when we consider the 2 pyruvates are entering in the Krebs cycle. And the third, we are talking about the ATP molecules. We have studied that the GTP was formed, that the GTP will also convert into the ATP. So in one cycle, Krebs cycle, there will be the formation of one ATP, and in the 2 Krebs cycle, there will be the formation of 2 ATPs. So the total products of Krebs cycle along with the pyruvate converting into acetyl-CoA, there are formation of 2 ATPs, 6 NADH, 2 FADH2 and 6 carbon dioxide molecule. I hope you all learnt about the Krebs cycle, that how this cycle is taking place in the mitochondrial matrix and how many products are forming here. So if you have any concern or any question about this cycle, you can talk in the comment section. And if you like this video, share this video with your friends. Thank you so much.